we can go ahead and address them for you. With that, I'd like to hand over to Dr. Pupak Farahani. She's our host for today. Thank you.
Hello, everyone. My name is Pupak Farahani, Marketing Manager at Advanced Cell Diagnostics. I'm your host for today. Welcome to ACD's Knowledge Resource webinar series, where we will give you closer understanding of how RNASCOPE technology is contributing to specific application areas. We begin today's knowledge series on a topic of applications using BaseScope assays, presented by Dr. Anushka Dikshit. Dr. Anushka Dikshit is an application scientist with Advanced Cell Diagnostics with background in cancer research and immuno-oncology. She is focused on expanding the applications of RNASCOPE technology for oncology research. Prior to working for ACD, Anushka was involved in identifying novel therapeutic targets for melanoma as a postdoctoral fellow at Duke University. She obtained her doctoral degree from Southern Illinois University School of Medicine in molecular physiology. With that, we'll get started. Anushka?
Thank you for the introduction, and welcome everyone to our presentation today, where I will be talking about our BaseScope assay and give you examples about how this technology has been used in different research areas to identify short targets, splice variants, and point mutations. So for those of you who are not aware of our RNASCOPE technology, it is an in situ hybridization platform designed specifically to identify target sequences or target transcripts without the traditional limitations of an in situ hybridization assay. So we have tweaked this technology to make it easy to use with high sensitivity and specificity, no background detection, and visualization of signal at a single molecule and single cell resolution. The great thing about this technology is that it is applicable to a wide range of sample and species type. And this technology has been used in a number of research areas to identify biomarkers in cells and tissues with morphological and spatial context. The three unique parts of this technology are the target probe design strategy, where we will design specific target probes against the target transcript of your interest. Then we have the signal amplification system, where we have developed a technology to ensure high degree of specificity and sensitivity by allowing high signal to noise ratio. And finally, our detection and quantification system allows you to visualize your target transcript as a single punctate dot at single cell resolution. As you can see here, we allow for either chromogenic or fluorescent detection based on your interest. And ultimately, we have the capability of semi-quantitative or quantitative analysis of this signal to support your research interests. As I mentioned, this assay is available in both chromogenic and fluorescent platforms, and we have been able to use this technology to not only detect traditional mRNA transcripts, but targets like long non-coding RNAs, microRNAs, siRNAs, ASOs, splice variants, short targets, and point mutations. Additionally, this technology can be made applicable in cells as well as in tissues. And this assay can be used on FFPE samples, fixed and fresh frozen samples, PBMCs, and cultured cells. In this slide, I'll be highlighting the two key features of our technology, which are the probe design strategy and the signal amplification system. Now, these two parts of our technology are what makes it stand apart from some of the other tools that are available for detection of target transcripts. As you can see on the screen, we depict our probes as Z molecules to highlight the different parts of this probe. The preamplifier binding site is the foundation of your signal amplification tree. The target binding site is where the probe binds to the target sequence of interest. And the way we design these probes are that we normally design them as pooled probes of ZZ pairs. As you can see, the two Z's or two probes have to bind in tandem next to each other on your target transcript of interest. And this forms a foundation for the preamplifier to bind onto the ZZ pair. To the preamplifier, the amplifiers bind on, followed by which you have your chromogenically or fluorescently labeled probes binding onto the amplifier. This sequential amplification strategy allows for building an amplification tree that gives you high sensitivity of detection, allowing even the lowest expressing targets to be detected using this assay. Additionally, we also ensure high degree of specificity. As I mentioned, the two Z probes have to bind in tandem next to each other on the target transcript for the preamplifier to bind on to the Z pairs. If you have a single Z probe non-specifically binding to another sequence, the preamplifier will not be able to bind onto this probe, and there won't be any amplification tree formed. The RNASCOPE technology encompasses three major assays. The RNASCOPE assay, which is for targets with above 300 nucleotides in length. And the probe design strategy includes designing approximately 20 ZZ pair of pooled probes that span on an average 1,000 bases of sequence on your target transcript. This assay is available in automated or manual formats, and we have flexibility of plexing that goes up to 12 plex. On the other hand, we have our two more niche assays that are your BaseScope and microRNAscope assays that are specialized for certain types of targets. For example, our BaseScope assay, which is going to be the focus of today's presentation, is mainly for detecting targets between 50 to 300 nucleotides in length. And we design the probes for this particular assay as either 1 to 3 ZZ probes. Again, this assay is available both in manual and automated formats, but currently we allow only one plex or duplex assays in this particular BaseScope technology. Finally, we have our latest launched microRNAscope assay, which, as the name suggests, is for even smaller sequences between 17 to 50 nucleotides in length, mainly for identifying targets like siRNAs, microRNAs, and ASOs. And currently, it's available as a one-plex assay in automated and manual formats. In this particular slide, I have detailed the extensive breadth of our product portfolio, just to give you a sense of how we have been able to develop our technology to identify different types of target transcripts and make this available in chromogenic and fluorescent platforms across manual and automated assays. So based on your target transcript of interest, you can pick any of these assays, and based on your plexing interest, you can pick which assay is most suited for your research. Currently, we have over 26,000 target probes in our catalog already that are pre-designed and ready to go. But if you're working with a unique target that we do not currently have a probe for, we can design the probe for you within 2 weeks. And as I mentioned, it can be any target in any species. So this type of flexibility really allows us to detect a large number of targets in different research and application areas. Some of the application areas are mentioned here on the screen, which includes detection of non-coding RNA, splice variants, specific CDR3 sequences on TCRs, GPCRs, antibody validation, combining RNAscope with IHC or IF, which I'll be talking about later in the presentation, and validation of high throughput assays like RNA sequencing. So because of this, we have been extensively used in neuroscience, stem cell and developmental biology research, for infectious disease research, mainly for assessing emerging pathogens, since we can design a probe as quick as 2 weeks to detect any novel and emerging pathogens. Then we have applications in biopharma discovery and development, especially to assess the efficacy of cell and gene therapies in preclinical models. And finally, to study tumorigenesis and tumor progression in immuno-oncology and cancer research. For today's presentation, I'll be touching upon multiple of these research areas while showing you the applications of our BaseScope assay. Now, our BaseScope assay is, as I mentioned, mainly for detecting smaller or shorter targets, namely splice variants, point mutations, and short targets. Now, why is it important to detect these small targets? Well, in oncology specifically, a lot of point mutations can potentially drive the initiation or transformation into cancer cells and also contribute in tumor progression. Similarly, certain splice variants are important when it comes to developmental biology or even oncology, where individual splice isoforms can have different functions, which can ultimately alter the functions of the actual cell or overall the tissue. And finally, short targets, for example, such as homologous sequences or CDR3 sequences, cannot be easily distinguished using the current tools that are available. And the BaseScope assay is a platform that not only allows you to study these highly homologous sequences or short targets, but gives you that spatial and morphological context where you can visualize these targets in your tissue of interest. So overall, using this technology, you can detect your target transcript of interest with high specificity and sensitivity, all the while retaining that morphological and spatial context. So overall, our BaseScope assay can be made applicable for detection of a large range of target transcripts. And these include your exon junctions or splice variants, which include exon-exon junction detection of certain circular RNAs, which are important in developmental biology, detection of gene fusions, or identification of certain knockout targets. Then you have your short sequence detection, where again, as I mentioned, detection of CDR3 sequences or highly homologous sequences in your target transcript of interest. And this also includes detection of certain pre-microRNAs, that is the premature form of microRNAs that can be identified using this assay. And of course, if you want to detect the mature form of the microRNAs, we have our newest microRNAscope assay that can be used. And finally, we have detection of point mutations, where we have validated probes to detect certain commonly occurring point mutations or point mutations that are most significant and implicated in certain solid tumor malignancies. And these can be quite tricky to detect in a spatial context, but we have optimized the probes for this and validated them in-house. So let's start by looking at a few examples of how this can be used for detection of splice variants. And in our first example, I want to talk to you about a very important application of this assay, which is to detect the splice variants or splice isoforms of the tau protein. Now, the tau protein has been implicated in a number of neurodegenerative diseases, including certain tauopathies, or Alzheimer's disease is an example, wherein the tau protein is aberrantly expressed. Now, the tau expression can be very significant with respect to the developing embryo, especially in the developing brain, wherein the different splice variants of these tau proteins have individual functions to play. But identifying these splice variants has been a quite challenging task for researchers that have been studying tauopathies or Alzheimer's disease. But using the BaseScope assay, we are providing a solution where you can individually identify these different splice isoforms of the tau protein and visualize their detection in situ. So the two major isoforms are the 3R tau and the 4R tau, wherein in the 3R tau you have the exon 10 exclusion, whereas in the 4R tau you have the exon 10 inclusion. And they play special roles in the developing brain since they have different functions. But aberrant expression of these splice variants can lead to certain neurodegenerative diseases. But in order to study them, it's important that we have a tool, a technology, where we can identify these splice variants and detect their expression. So using the BaseScope assay, we were able to design probes that specifically span the exon 6, exon 7 junction or exon 9, exon 11 junction to detect the 3R tau variant. Whereas to detect the 4R tau variant, we would identify them using the exon 6 or exon 7 or the exon 9, exon 10 junction. So the two main probe pairs that differentiate between the 3R and 4R variant are these two, the ones marked in yellow and blue. Since the 4R tau has the exon 10 and the 3R tau has the exon 10 repeat missing. As a result, the 911 probe will only identify the 3R tau, the 910 probe will only identify the 4R tau, whereas the 67 probe will identify both the 3R and 4R tau. And here is an example where we have used the BaseScope assay in combination with immunohistochemistry detecting mu-N, which is a pan-neuronal marker. And we're focused on the brain cortex region of the mouse. And we are looking at 4 different developmental stages: day 0, day 10, day 30, and day 56. And I want to show you the differential expression of the 3R tau variant in these developmental stages, wherein you can see the nice red punctate dots abundantly expressed in the day 0 embryo here. But as you go up in developmental stages, the expression of this 3R tau decreases. Similarly, when we look at the 4R tau variant, you can see the expression in day 0, but the expression increases dramatically at day 10, and followed by which at day 30 and day 56 again goes down to those of levels at day 0. So the expression of the 4R tau as against the 3R tau seems to be reaching its maximum level at day 10 of the developmental stage. So this here shows you that the roles of these individual splice variants can be quite different based on the developmental stage. Here I have compared the hippocampus area of the mouse, and we've compared the day 0 of day 0 of development, and we are looking at the expression of the 3R and 4R variant side by side. Again, the expression seems comparable between these two splice variants at day 0. Now, as we go ahead in the developmental stage at day 10, as we saw before, even in the hippocampus, the expression of the 4R tau variant is dramatically high compared to the 3R variant, where the expression seems to have dropped significantly. Moving on to day 30, again, expression seems to be higher in the 4R tau, but compared to day 10, the expression in day 30 is still lower. And then day 56, again, expression for 4R seems to be higher, but not as high as it was on day 10. So it shows you the differential expression in different parts of the brain for these 3R and 4R tau variants, and potentially helps you better understand the function of these different splice variants in the developing and postnatal brain. So overall, what this study showed us is that there is a gradient of expression based on the developmental stage for both the 3R and the 4R tau variant, wherein the 3R peaks at day 0 and goes down, whereas the 4R tau variant peaks at day 10 and then goes down from there. So this differential expression of the 3R and 4R tau variants can be important to study in these neurodegenerative diseases and potentially help identify the expression pattern and the role of these splice variants in these degenerative diseases, including Alzheimer's. So using this BaseScope tool, we have really provided a solution to researchers studying neurodegenerative disorders and especially tauopathies to differentiate between these tau splice variants. Next, I want to show you an example, again, in neuroscience research, wherein the BaseScope assay has been used to visualize a excised transcript for Reb1 target. In this particular example, the researchers are studying the expression of Reb1 mRNA and its role in promoting MTOR and its role in promoting MTOR activation in tumorigenesis. So this is an example in cancer research. And here we are using the BaseScope assay to differentiate between the intact transcript of Reb1 and compare the expression to an excised or modified transcript of Reb1 in the mouse model. So what they want to show is that MTOR is known to promote tumorigenesis and tumor progression in multiple solid tumors. And here they are studying the role of MTOR in breast cancer tumorigenesis. It is well established that the GTPase Reb1 promotes or activates MTOR expression. And in this particular example, they have used a mouse model where they have ablated Reb1 mRNA by using a knockout model, wherein the knockout model produces an Reb1 transcript that is excised and non-functional. And using the BaseScope assay, they were able to visualize the intact transcript and the excised transcript in the wild type animals and the knockout animals. So in this figure B, you can see using the junction probe, which is the probe to detect the ablated Reb1 transcript, they were able to show the expression of this transcript in the floxed or knockout mice and no expression in the wild type mice, as you would expect. But when you visualize the intact transcript, the expression is present in the wild type mice and none in the knockout mice. So this is exactly what you would expect. And the red punctate staining, as you can see, is our BaseScope staining. An important thing to highlight here is that the BaseScope assay is chromogenic, but the red chromogen that we use is fluorescent. That means when you visualize a red stain under the microscope, under the fluorescent microscope, it fluoresces in the red channel. So this is a very important, advantage of using the red assay, since it allows you to visualize your target under the fluorescent microscope if you want to get that really good contrast. So ultimately what they found was when they ablated the Reb1 transcript, there was a significant delay in tumor development in this breast cancer model, where ablation of Reb1 delayed tumor initiation, wherein the wild type model developed tumors at 125 days, the Reb1 knockout models developed tumors at 375 days. So significant delay. Additionally, what they found was when they combined the BaseScope assay with immunofluorescence for detecting KRAS, they found that the excised transcript, as expected, was expressing at a higher degree in the knockout model. But what was important was they saw a nice correlation between the KRAS expression and the Reb1 excised transcript detection, showing that when KRAS was specifically expressed in the epithelial cells of the mammary glands, that's where you saw the excised transcript expressed. On the other hand, when they looked at the intact Reb1 transcript, in the knockout model, they saw that the intact transcript was not correlating at all with the KRAS expression, which means in cells that were expressing the KRAS, they were not seeing the intact Reb1 expression, indicating that those are the cells that will potentially have the knockout or excised transcript expression. And they have done a very good job quantifying this, showing the correlation of the knockout model to expression of the excised transcript, as you would expect. And this model really helped them study the expression of the excised transcript and determine the role of Reb1 in MTOR activation. And ultimately, what they were able to show was that Reb1 was in fact vital for MTOR activation, but in the absence of MTOR, in the absence of Reb1, MTOR itself started acquiring hyperactivating mutations and promoting to breast cancer tumorigenesis. So this technology was extremely useful for them to develop an animal model where they could establish the role of MTOR in tumorigenesis. In this next example, these researchers were studying endometrial cancer, which is a type of endometrial cancer, and they were wanting to identify a molecular signature for specific subtypes within these endometrial cancer. So some of these subtypes are known to have really poor prognosis or really good prognosis, but then there are these two subtypes called the MMRD or P53 wild type subtypes that have sort of an intermediate prognosis, which means it can basically be either good or bad based on the patient. So to have a more clearer picture for risk stratification, it's important to identify more molecular markers that would make the prognosis more clear. So they turned to this target called FGFR, which they learned that FGFR 2B to 2C switching isoform switching is found in a number of solid tumor malignancies. So what is the role of this FGFR2 switching in endometrial cancer? So for this, they wanted to visualize the expression of FGFR2C and determine its prognostic value in these endometrial cancer tumors. So using the BaseScope assay, they were able to design specific probes against the 2C isoform and the 2B isoform to differentiate their expression. And this is a great example where they've compared the expression of 2B and 2C in two different cell lines. The Ishikawa cell line, which expresses the 2B isoform, and the AN3CA cell line, which expresses the 2C isoform. So as you can see, using our BaseScope assay and specific probes targeting the 2B, detection was only seen in the Ishikawa cells and none in the AN3CA cells. Vice versa, the 2C isoform was only detected in the AN3CA cells and not in the Ishikawa cells, highlighting the specificity of our BaseScope probes and identifying these splice variants. And again, here they have done IHC for the FGFR2 protein, highlighting that this particular IHC was not able to differentiate between the isoforms, whereas the BaseScope assay was. So they did extensive BaseScope analysis in over 400 samples to study the expression of the FGFR2C isoform in these samples. And they went a step ahead and did our semi-quantitative scoring for these samples to establish the degree of expression in the different endometrial cancer samples. And what they were able to see was that 302 samples that they identified were actually, that showed the expression of FGFR2 were actually endometrial type of samples, and 84 were non-EEC type of tumors. And about 50% of these EEC tumors had the C form of FGFR2 expression, and only 23% of the non-EEC showed the C form of FGFR2 expression, which means that the isoform known to promote cancer, the C form, was getting expressed in the EEC type of tumors. Since they did scoring, they were able to establish that over 15% of the EECs that showed FGFR-C expression had high FGFR2C expression, with score of either 3, 4, or plus, indicating that high degree of expression of the CFGFR2 isoform was seen in the EEC type of samples. So this allowed them to show that the C form of FGFR2 in turn was playing some role in promoting the EEC tumor growth. And they went a step ahead again and compared different grades of these EEC tumors. And what they found was the grade 3 or the higher grade of tumors showed even higher expression of the FGFR2C isoform, again indicating that it could potentially be a good prognostic marker to show poor prognosis and overall poor disease-free prognosis in these endometrial cancer tumors. So using the BaseScope assay, they were able to analyze samples and establish a prognostic value for FGFR2C isoform in these tumors. Next is another example where this was used to differentiate between the splice variants. Our BaseScope assay was used to study alternate splicing of SMAD4. Now, SMAD4 is an important signaling molecule, as you all know, but plays a special role in myogenesis. Circular RNAs do have specific regulatory functions, and the SMAD4 circular RNA plays a role in myogenesis, although the role of SMAD4 is not very well defined. So in this particular paper, they wanted to observe the expression of the linear SMAD4 and the circular SMAD4 in these differentiating myoblasts. So what they were able to show using our duplex assay was they were able to visualize both the linear and circular form of these SMAD4 target in these tissues. And what they were able to show that in the differentiating muscle cells, they were able to show an increased expression of these the linear form of SMAD4 mRNA, showing that the alternate variant of the SMAD4 did play a role in myogenesis. So by comparing the linear and circular form, they were able to shed some more light on the myogenic differentiation and the role of SMAD4 in it. Next, we're going to look at an example of how this assay was used to detect point mutations. As I mentioned, within ACD, we have several different point mutations that we have validated in-house. And these are your key targets like KRAS, BRAF mutations, EGFR, and PI3K, which are most commonly found in a number of malignancies and are of interest for researchers. So we have validated these in-house, and we know these probes are specifically able to identify target point mutations. So I'm going to go over a study that we did recently for the AACR 2020 meeting. And here what we did was we wanted to identify different KRAS point mutations in a 48-core lung cancer tumor TMA. And we did this by using the BaseScope Ventana automated assay. So for this, what we did was we used 4 different KRAS point mutation probes, detecting either specifically the G12C mutation, G12S mutation, G12V mutation, or G12A mutation. And we also had 2 different control probes that detected the control or wild type KRAS transcript. And we visualized the expression of these transcripts on 48 different lung cancer samples. And what we found here was, just giving you an example, how the signal appears using our point mutation probes. Now, these are 2 probes from the TMA, core 8 and core 5, which have wild type BRAF expression. And so using our wild type BaseScope probes that are similar in design to our point mutation probes, we were able to detect nice punctate dot indicating wild type detection or wild type BRAF expression in these cores. And next, we went on to detect the G12A and G12C mutations. Again, this is core 41, which is known to have the G12A mutation by DNA sequencing. Similarly, core 39, known to have the G12C mutation verified by DNA sequencing. And using our BaseScope assay, we were specifically able to identify these cells that were expressing these mutant KRAS cells. I'm sorry, mutant KRAS transcript. Again, an example of how we use specific probes against the G12V mutation and the G12S mutation to identify these mutant positive cells within these tumor samples. And an important thing to note here is, if you see for some of these samples, the expression of these point mutations is limited to a subset of these tumors. So really highlighting the fact that using the BaseScope assay, we were able to identify these clonal, subclonal populations of cells within this tumor samples that sometimes might be lost by using bulk analysis techniques like DNA sequencing. So overall, we did a quantitative analysis on these samples. And what we were able to show was compare our BaseScope assay data to that of the mutation status obtained by DNA sequencing data. And what we observed was our data was in correlation to that of the DNA sequencing data that we obtained, showing that we had high degree of sensitivity and specificity for all these different point mutation probes. So showing that BaseScope, if compared to DNA sequencing, showed 100% sensitivity and specificity for G12C probe, 100% sensitivity and 97% specificity for G12A probe, about 83% sensitivity and 100% specificity for G12V probe, and 100% sensitivity and 97% specificity for the G12S probe. So overall, showing that our data was concordant with that of DNA sequencing and can be used as a validation tool for point mutation detection, all the while providing the spatial and morphological context. So this data really showed us that we are on par with DNA sequencing when it comes to detecting point mutations. Unlike the sequencing methods, which are bulk methods, we're able to identify those subclonal populations of cells, which might be very few within the whole tumor sample. And this is really our strength to identifying these cell types using the BaseScope assay. This data was presented at the AACR meeting and was very well received. We also have this poster available on our website, acd.com. So next, the final application of this assay is to detect short targets or highly homologous sequences. And an important example of this is for development of cell and gene therapies. And in this particular example, we did a study for Ultragenyx, where they're developing an AAV-based gene therapy. And here, without disclosing the target, we have shown that our BaseScope assay duplex was used to visualize the target transcript, that is the transgene, and the AAV vector DNA in this transduced cells. And we compared that to a vector-treated, or vehicle-treated sample. So what you can see here is cells that were transduced by this AAV vector and transgene. And using the BaseScope assay, you can detect both the transgene and the vector. Now, what I want you to appreciate here is the subcellular resolution that is afforded by our assay, where you can see the individual nucleus and cytoplasm. And you can see that the green signal, which highlights the vector genome, is specifically seen in the nucleus of these transformed cells. But the transgene is expressed in the nucleus as well as in the cytoplasm, as you would expect, since it's a transgene mRNA. So this application really shows you that we can go down to not only single cell, but subcellular resolution, and allows you to visualize the biodistribution and expression of your transgene while testing preclinical models for your gene or cell therapies. Again, we were able to quantify this to show you the degree of cells that were transduced by this therapy and count the number of positive cells for individual transgene and for vector transduced cells. Next, I want to highlight an application of the whole RNASCOPE technology overall, which is to validate single-cell RNA sequencing data. In the last few years, this technique has really come out as a technique that allows you to go down to single-cell resolution to but detect gene expression data at that single-cell level. But then what you lose out is on that spatial, morphological, and tissue context. So a number of researchers like to perform RNA sequencing on target tissue of interest and then validate their cell populations using our RNASCOPE or BaseScope assays in situ. And now I'm going to show you an example of how the BaseScope assay was used as a validation tool for single nuclear RNA sequencing in this particular paper. This paper published in Nature was studying MS, multiple sclerosis, and were trying to compare the differential expression of certain specific oligodendrocyte populations within the healthy brain and the MS brain post-mortem. So in this particular image, they were first wanting to validate their single nuclear RNA-seq data by using specific BaseScope probes against markers that identified oligodendrocytes. So they identified different individual subclusters of oligodendrocytes, and using the BaseScope probes were able to identify these subtypes in situ. So using these KLK6 and CDH20 probes, they were able to identify oligo5 and oligo1 population respectively. Similarly, using opalin and LURAPL1, they were able to identify oligo6 and oligo2 subpopulations. So when they further compared the single nuclear sequencing data, they were able to show that there was certain differential expression between the normal healthy brain and the MS brain when it came to certain specific markers. And these were markers like olig1, 2, and CD74. Now, these are markers for microglial cells and oligodendrocytes. And what they were able to identify was that there are certain cells called oligodendroglial cells which have hybrid characteristics of both oligodendrocytes and microglia, and therefore express markers for both the oligodendrocytes and microglia. And using the BaseScope assay in combination with IHC, they were able to show that these hybrid cells do exist and were able to visualize these hybrid cells in situ. Additionally, they were able to show that the MS cells or the MS brain show a high degree of these hybrid cells compared to the normal brain, indicating that the presence of these oligodendroglial populations might be contributing to the MS pathogenesis. Next is an example of how our assay can be used to detect short sequences, such as premature microRNAs. In this example, they are studying the role of microRNA 135 in cancer progression. So gastric cancer is known to arise from inflammatory gut tissue. So they're trying to establish the role of chronic gastritis in promoting gastric carcinogenesis. And in a chronic gastric tissue, the cells are exposed to high degree of inflammatory cytokines. And this might play an important role in malignant transformation of these gastritic cells. What they have shown is that certain microRNAs can play an important role in this transformation from normal to carcinogenic, normal to malignant cells, but they've not been able to establish specifically a subset of microRNAs that might be playing this role. So in this particular example, they were studying the role of microRNA 135P in malignant transformation of gastric tissue. So they used our BaseScope assay to design probe against the premature form of the microRNA and study its expression in either wild type tissue or in gastritic tissue or in gastric cancer in this mouse model. And what they were able to show is that in the wild type tissue, there is very low to no expression of MIR135P, but in gastritis and in gastric cancer tissue, you see high expression of 135B, in turn suggesting that this microRNA might have some role in malignant transformation of gastric tissue. Now, our BaseScope assay is very successful in detecting premature microRNAs, but we have our microRNAscope assay to detect the mature form of these microRNAs if the researchers are interested. Another example of how premature microRNAs can be detected using BaseScope assay is this example of studying cocaine-associated psychiatric behavior in a mouse model, where they established the role of MIR135P in cocaine-induced addiction. So MIR134 is expressed at a high degree in the hippocampus, and what they observed is that it might have some role in promoting anxiety and depression type behavior in cocaine addiction. So using this mouse model, they were studying the expression of 134B and its role in promoting this psychiatric behavior in cocaine addiction. Now, 134B is known to play an important role in overall dendritic spine health, glial activity, and maintaining neurochemical microenvironments. But under cocaine addiction, this is disrupted, and the expression of microRNA 134B is seen to be increased under in these mice with cocaine addiction. So in this particular example, they have combined BaseScope assay with immunofluorescence to show cell type specific expression of MIR134 in hippocampus, which will ultimately provide clues as to the exact function of MIR134B in promoting cocaine addiction. So what they were able to show here is the expression of 134B in different parts of this hippocampus, and show the expression in combination with different microglial and neuronal cell markers, suggesting or identifying the specific cell types in which 134B is expressed. And overall, they were able to show that MIR134B was expressed in different parts of the hippocampus, but predominantly in some parts that is high in the ventral part of hippocampus compared to the other parts. And this beautiful image was generated, as I said, using BaseScope, combining that with immunofluorescence, owing to the fact that our chromogen can fluoresce under the fluorescent microscope. So again, this assay was used to detect the premature form of 134B. Our newest microRNAscope assay can detect the mature form of this particular target. So we have realized that our researchers are really interested in combining the BaseScope or RNAscope assays with immunohistochemistry and immunofluorescence, allowing co-detection of RNA protein targets. And as a result, we wanted to make this process feasible for our researchers, and therefore we recently launched our co-detection assay kits, which allows you to easily combine your RNA and protein RNA and BaseScope. RNAscope and BaseScope assays with either ISH or with either IHC or IF. So this will allow you to detect your RNA and protein targets simultaneously on your target tissue of interest. And the applications of this particular workflow are endless, where it can be used to visualize pathogen infection and detect host cell markers, or visualize certain non-coding RNA targets in specific cell types, or as you saw in the previous example, detect your microRNA targets in specific cells within tissues of interest. And so we wanted to make this process really easy for our customers, wherein you would not have to compromise on your IHC or IF signal because of our RNAscope or BaseScope workflow. And to that effect, we launched this kit, and we have included three major reagents: the co-detection blocker, the antibody diluent, and the target retrieval reagent, which you would use to, which in combination with a new integrated workflow will give you the most optimum RNA protein co-detection. This we have made available with a range of our BaseScope and RNAscope assays as seen on the screen. Now the workflow for this particular assay is a little bit different than a traditional sequential ISH-IHC workflow, where you will permeabilize your cell, and before you perform your ISH, you will add your target antibody of interest and bind your antibody to your target protein, followed by which you will perform your ISH assay and then amplify your ISH assay, detect your antibody signal, and then visualize both RNA and protein. So instead of performing your IHC after ISH, we have tweaked the workflow such that now you will perform your IHC before you perform your ISH, and thereby ensure no loss of your IHC or protein signal. And this enables you to combine your target antibody of interest with your target RNAscope or BaseScope probe of interest. And this has really allowed researchers to encompass a much larger range of antibodies that are now compatible with our RNAscope and BaseScope assays. As I mentioned, the three different reagents will be used based on which assay you're using. The Leica assays or the automated assays currently require the use of only our primary antibody diluent for this particular workflow. Now, I just want to highlight a few examples of how effective this assay is. I have compared IHC only data with our sequential or HYBRID sequential or traditional workflow and compared that to our new co-detection or integrated workflow. And some notorious antibodies like CD8, which would not be compatible with the HYBRID workflow, are now compatible with the new co-detection workflow, where you can see we have rescued the CD8 signal in this third panel. Some antibodies like CD3 were originally compatible and are still compatible with the new workflow. Additionally, this assay, as I mentioned, is not only compatible with our BaseScope, but also with our RNASCOPE assays, the chromogenic red assay, as well as the BaseScope red assay. This assay is also compatible with our multiplex assay, where here we have combined the Ki67 antibody with our RNASCOPE control probes, showing you optimum detection of both RNA and protein targets. Again, example of some notorious previously incompatible antibodies like CD8, CD20, CD19 are now compatible with robust detection of your protein signal with your RNASCOPE targets. So with this particular workflow, we really hope that the researchers can choose their favorite antibody of interest and combine that with their RNASCOPE or BaseScope targets. And here's a broad overview of how this assay is compatible with different RNASCOPE and BaseScope assays and what reagents you would need to use this workflow in combination. Please reach out to us for more information about this or visit our website. So overall, this HYBRID workflow will really augment our RNASCOPE assay in providing that multi-omics approach where you can detect your RNA and protein targets simultaneously on the same section. Since we've made this available with both our chromogenic and fluorescent assays and both our manual and Leica platforms, a number of researchers will be able to use this for their particular projects. Overall, our RNASCOPE and BaseScope assays have been very well adopted. We have almost 3,000 publications now in different research and application areas. And today, I hope I have shown you that our BaseScope assay is key in detecting those unique small RNA targets like homologous sequences, short targets, point mutations, and splice variants that fall between the range of 50 to 300 nucleotides. We make this assay available in both chromogenic and fluorescent platform, in both automated and manual platforms, and we can visualize our chromogenic assay under the fluorescent microscope to detect our red chromogen as a fluorescent signal. And finally, as I mentioned, our assay can be combined with our immunofluorescence or immunohistochemistry assays using our new co-detection kits. So with that, I will take any questions.